Hello everyone. This is a brief discussion of our paper titled Brain Functional Connectivity as Biomarker for Propofol-Induced Alterations of Consciousness. I'm Anusha AS from the Medical Intelligence and Language Engineering Lab at the Indian Institute of Science, Bengaluru. The presentation coverage involves a brief introduction of the problem. Then we will proceed to the description of the data set, followed by methodology of analysis, results, discussions, and conclusions. The study investigates EEG-based functional connectivity as an objective marker for the reversible altered conscious states induced by propofol, an anesthetic drug commonly used in clinical medicine. We further attempt to incorporate EEG-based FC measures into machine learning algorithms to establish a classification scheme that can distinctly identify altered conscious states during anesthesia. The motivation for the study is, of course, the fact that EEG is relatively economic and easy to measure from the scalp. Also, it has long been known to implicate anesthetic-induced changes in brain dynamics. Even with these advantages, an EEG-based depth of anesthesia monitor is still not widely used in clinical settings. This is despite the fact that intraoperative awareness during anesthesia continues to result in pain and prolonged and unwanted outcomes like post-traumatic stress disorder or depression in patients. This makes a robust EEG marker which can reliably track the loss and reestablishment of consciousness during anesthesia, an important and useful clinical requirement. For this study, we used the propofol sedation database available for public access in the University of Cambridge data repository. The dataset has EEG recorded in 20 healthy participants of age 30 and standard deviation 11. They were given the sedative propofol. For each participant, there are 4 data sets, each with approximately 7-minute duration, recorded at baseline during mild sedation when the propofol concentration measured in blood plasma was 0.6 microgram per milliliter, moderate sedation where the propofol concentration in blood plasma is 1.2 microgram per milliliter, and finally at recovery. Data preprocessing involves filtering EEG time series from 21 channels as shown here in the figure between 0.5 to 45 hertz, segmented into 10-second epochs, cleaned and preprocessed to remove artifact and referenced to the average of all channels. The labeling was done based on the experiment phase. We further proceeded to the functional connectivity estimation. The debiased estimator of squared weighted phase lag index was used as a metric for functional connectivity computation. The metric computes phase synchronization between signals. The computation considers only the imaginary component of the cross spectrum of 2 time series and is not largely affected by the volume conduction artifacts. The mathematical expression is as shown here. The Im of x denotes the imaginary component of the cross spectrum of complex valued random variable x, and n is the number of trials. So basically the estimation involves computing imaginary components of the cross spectra, then computing the average imaginary component of the cross spectra, and further normalizing it by computed average over the magnitudes of the imaginary component of cross spectra. Now, functional connectivity between all possible pairs of EEG electrodes was computed using the FC metric, resulting in a feature vector of dimension 210 for each epoch. The computation was done separately for 5 EEG subbands, namely delta, theta, alpha, beta, and low gamma. Further, Baruta-based feature selection was done separately for each EEG band to identify all appropriate functional connections in each band. The Baruta selected connections were further used as features to develop a random subspace ensemble frameworks for classification. In random subspace ensemble, as the name suggests, the training data is modified such that random subspaces of feature space are chosen and classifiers are built for each subspace. The result of these classifiers are further aggregated by utilizing a majority voting or weighted average. The scheme actually benefits from the aggregated decision of such classifiers and provides better predictive results than probably a single classifier built on the original training set in the entire feature space. We can tune the base classifier to be used, number of iterations, seed and subspace size to yield the best performance. The performance index chosen was 10-fold cross-validation accuracy. Moving on to results, what is shown here in the left is the whole brain connectivity in 5 EEG bands, namely delta, theta, alpha, beta, and low gamma during baseline, mild, and moderate sedation, as well as recovery stages. The assessment was done by averaging FC values of all 210 connections across multiple epochs for each band during specific stages. Also, a pairwise comparison of means was performed using the Bonferroni method to determine if differences are statistically significant, and only the statistically significant differences are highlighted in the figure, with 1 star having a statistical significance of 0.05, 2 showing it to be at 0.01, and 3 at 0.001. So we can see that the alpha band functional connectivity is significantly higher during baseline, mild, and moderate sedation, as well as recovery stages. Also, as the sedation level increases from mild to moderate, the connectivity in the alpha band decreases, although the decrease is not statistically significant. However, the alpha band connectivity increases significantly as the subject proceeds to the recovery stage from the moderate sedation stage, indicating that the alpha connectivity is indeed compromised during sedation. This observation warranted a more detailed investigation of connectivity in the alpha band, which is what is presented on the right. So this is the grand average alpha band connectivity map of 20 subjects during 4 stages of sedation study. From the map, we can see that the loss of consciousness during moderate sedation is accompanied by a decrease in the cortico-cortical connectivity from frontal to parietal and occipital regions of the brain. This in turn has contributed to the overall reduction in the whole brain connectivity. Also, we can see that these connections are more or less preserved during mild sedation. We can conclude that there is a reduction in the efficiency of frontoparietal and fronto-occipital networks during moderate propofol sedation. As already mentioned, Baruta-based feature selection was done separately for each specific EEG band to identify the all relevant feature subset that can effectively distinguish the target classes. The connections identified as significant is shown in the table. There are 18 connections identified as significant, which involved electrodes in the frontal lobe of the brain. So in the 17 out of these 18 connections used frontal electrodes, and also a total of 7 connections belong completely in the frontal region. That is, both electrodes belong to the frontal lobe. These we think may be an indication of an association between frontal brain connectivity and propofol-induced unconsciousness. Also, 2 connections along the anterior posterior axis of the brain, namely the FPZ-P3 and FPZ-O2, which can be considered as long-range connections, were identified as significant in the alpha band. This is in line with the previous evidence highlighting the inhibition of frontal to parietal and frontal to occipital connections during general anesthesia. The Baruta selected connections were further used as features to develop RSE frameworks for classification. The performance measures of classification models developed using features from specific EEG bands as well as their concatenation are summarized here in the table. As can be seen, the highest cross-validation accuracy of 98.75% was achieved while using all 18 features spread across 5 EEG subbands. If you observe the alpha band results, it will show that the learning model developed using only alpha band features can distinguish the moderate sedation and recovery phases reliably. This is also of significance in the context that the sedation safety standards put forth by the Association of Anesthetics of Great Britain and Ireland has identified transition from sedation to recovery as a period of high risk to the patient and has recommended continued monitoring until the patient fully recovers. So classification accuracy of the reported order yielded by the proposed model is an indication of the utility of functional connections across the human brain as characteristic signatures and potentially reliable correlates of conscious states associated with propofol sedation. The fact that the proposed classification framework utilizes only 18 functional connections involving 14 EEG channels is also advantageous in a clinical setting. Reduced number of electrodes can make the system less cumbersome. Also, a majority of these electrodes are in the frontal lobe, and it might allow for a rapid placement of electrodes below the hairline. Also, a learning model developed using only the alpha band features can distinguish the moderate sedation and recovery phases reliably. Thank you.